So I'm Evan Smith. I'm
You want to hold it?
Yeah.
Then I don't have to.
I'm a biology major at the University of Notre Dame, going into my junior year. This summer, I've been working with the Fei Wei Li lab, and specifically, I've been studying hornworts.
Perfect. So the purpose of your project and maybe the overall significant findings, was there anything that was really exciting for you in your project here?
Really exciting just overall, it's been a great introduction to like plant genetic engineering, which is a future career that I'm looking into. So just getting to learn from my mentor, specifically this Biolistic transformation protocol. It's been really cool. So
Can you explain that a little bit more?
Yeah, so this summer I've been working with hornworts, and basically they have these two really cool features. They have this carbon dioxide concentrating mechanism, which increases the efficiency of photosynthesis. And they also have a symbiotic relationship with cyanobacteria. So we're interested in studying like the genetics responsible for these features. So I've been working with this Biolistic transformation method, which is a way that we can genetically engineer the plants. And so overall this summer, I've been trying to improve the efficiency of this protocol.
Right. And what did you find? Did you improve it?
Well, yeah, I tested 4 different variables. We've been using this protocol for a few years now, so it's already working pretty well. But I tested like the shooting distance, so like how far the plasmid construct is from the cells. I also tested like the days post homogenization, which is like how long we allow the cells to grow before we transform them. I also tested an alternate DNA precipitation method, and lastly, using a different amount of DNA. And so overall, the results kind of just confirm the optimization of our current protocol. I didn't find too much that was interesting, but I got to test a lot of different variables, different ways of doing the protocol. So.
It is interesting to me when I was looking at it earlier, the shooting distance, it has to be pretty, like it didn't get better when it was shorter. Is there any reason you can hypothesize for that?
Yeah, that can be due to tissue damage, because as everyone knows, gold is very dense. So when you fire the gold at the hornwort cells, sometimes they can die instead of actually, expressing your DNA of interest. So.
All right. Wow. Yeah. Not ideal. You don't want to kill your test subject. All right. Is there anything else that you would do beyond this to try to optimize the whole procedure? Is there anything that going forward that you would want to try if you had more time?
Yeah, definitely. I'd love to increase my sample sizes, because if you look at some of the statistics, they're not great. Not quite, significant in terms of just the p-value. Yeah. But then, yeah, exactly. But I'd also like to test maybe using more gold in the protocol, which maybe the DNA has a better chance of coding onto. And then there's, of course, other methods for precipitating the DNA that I'd like to test as well. So.
Well, thank you so much, Evan. I hope that you had a lot of fun with this. It seems like you were busy and you had a good time. So, well, thank you for sharing with us.